Hi, I'm Charles Sabine, and this is Palm Springs. A record number of pharmaceutical companies, 60, are here for the Huntington's Disease Therapeutics Conference. They heard about pivotal developments in every avenue of research, not least regulatory authorities agreeing ways to speed up HD trials.
We asked them, can we use cUHDRS, the composite score that measures not just function, but also motor symptoms and cognition? Can we use that composite score as a primary endpoint to support an accelerated approval? And they said yes. And then we asked them, can we use data from the current studies, the ongoing studies, one in the US and one in Europe? Can we combine them together and will they form the basis for approval? And they said yes. We asked also one more question. Can we use the change from baseline in NFL that we've observed after 2 years, which in our case was the very first time we demonstrate a reduction from baseline in CSF NFL that is statistically significant in our study? And they said, yes, we agree that this could be used as supportive to your trial. So those were really massive positive responses that we got from them. The study is ongoing and we continue to accumulate data. We saw that after 2 years of treatment, patients who look like ours in our trial would have progressed by 1 point on cUHDRS, whereas those in the high dose in our trial, the 9 in the high dose, only dropped by 0.2. So one would say, okay, so what does that 0.8 mean? 0.8 out of 1 is about 80%. That's significant. That was the first time that we truly had statistical significance. Slowing the disease by that much is going to be impressive. And this has never been shown before in Huntington's disease, and it's very exciting for us to see.
And anything that shows a slowing down of the progression of the disease is revolutionary.
Yeah, I agree. So now that we've had the meeting with the FDA back in November, the next step is to go into some technical details around the manufacturing of our product, as well as the statistical analysis plan. And those meetings are going to happen within the next couple of months, after which we will communicate externally the outcome of this. In addition, we started discussions with the European regulatory agencies to also try to bring this medicine as quickly as possible to European patients as well.
Everyone benefits.
Exactly.
One of the other amazing successes of your work with the FDA was that they've also approved NFL as a biomarker, isn't that right?
Well, that's correct. It's called neurofilament light chain. It's something that exists inside the neurons. It's very sensitive. Whenever neurons die, for whatever reason, it leaks out. It leaks out into the CSF, and part of it leaks out into the plasma as well. So when you look at our data, when we show for the first time in a trial that after 2 years, NFL levels decrease statistically in our patients from baseline, that's an indication that the level of neurodegeneration in the brain has decreased, because you would expect that the levels of NFL increase by about 10 to 15% per year. So after 2 years, there should be about 25 to 30% increase. In our case, we had about 11% reduction from baseline. So this is very clear indication that you are somehow reducing the ongoing neurodegeneration that happens in Huntington's disease, and it cannot be but an objective measure of this improvement in effect. So you to some degree, you're saving neurons. And I think the agency agrees with this, and they think that this is a good supportive line of evidence in addition to our clinical data.
And PTC Therapeutics has also got fast-track approval from the FDA for its small molecule drug PTC 518, now renamed Votoplam.
What comes now then in the next 6 months?
In the next 6 months, we will be able to reveal the entire data set for all patients who participated in Pivot HD. So that 1-year trial, all of that data will read out. And that's an important milestone for us in the development of Votoplam. So it's one thing to look at a cohort of the first 30 subjects. It's another thing to look at hundreds of patients who have participated in Pivot HD to ascertain the effects of Huntington lowering on a Huntington disease population.
And that data will be released, around July time?
We said second quarter of 2025. Around that time is when we expect to have that full data set for public consumption. 2024 was a very big year for PTC. In June of 2024, we were able to look at an entire 12 months of patient data. But uniquely in this data release, we released the Huntington lowering in the CSF. Because PTC 518, now Votoplam, is administered orally, it's distributed uniformly throughout the body, and we can measure the amount of Huntington protein that's being lowered directly from the blood. we were able to show that we had synchronous HTT CSF lowering as we did in the whole blood.
Right.
Truly remarkable. It's really important in this particular trial to show that correlation because nobody's ever done it before.
People who follow this will have read a lot about Novartis and PTC. Novartis is one of the biggest 5 companies, pharma companies in the world.
That's right.
What's going on here with Novartis and PTC?
In the latter part of last year, in 2024, Novartis and PTC entered into an agreement, which is a licensing agreement for the acquisition of PTC 518 or Votoplam. Now we have this partnership with Novartis that has, as you described, a large cadre of resources to draw from in deployment for the larger phase 3 trial and beyond. We have a partner now that has also been in the oral splicing modifier space in Huntington's disease. So our collaborative experience over the last several years is now united toward this common cause of bringing an oral splicing modifier forward for the treatment of Huntington's disease.
So what's exciting about the update was the first demonstration ever of allele-specific silencing in patients. These data were incredibly important in that they demonstrated substantial lowering of mutant Huntington protein, so 46% lowering of mutant Huntington, but for the first time demonstrated that we could do that without reducing the healthy protein in the brain. These data were also important because we showed the first ever correlation between that reduction of mutant protein with slowing of caudate atrophy.
Atrophy means
decreasing in size, losing it.
Like shrinking.
Shrinking. So if we think about the underlying pathology in Huntington's disease, what happens is there's a atrophy of the caudate. That's a part of the brain that's responsible for a lot of the motor symptoms. If we can slow that atrophy in caudate, you can delay the progression of the disease. So it really demonstrates now the fact that mutant Huntington lowering in the setting of wild type sparing could have anatomical benefit for patients. What's really unique again about caudate as a biomarker is it really is the source of where the disease is occurring. So if we can demonstrate as a biomarker that you're slowing that rate of atrophy within the caudate, it means we should be having an impact at the site of where the disease is occurring and can correlate that with additional changes. So a change on MRI, but you can correlate that with a really important outcome, total functional capacity, so this delay in disability.
That functional capacity refers to people actually literally things like their movement and their ability to multitask.
Exactly. The ability that robs people of their function, right? That change in total functional capacity means that people are able to function longer, work longer. And in a fact analysis that we did showed that a 1% change in caudate atrophy could correlate to a greater than 7.5-year delay in disability on total functional capacity. So these are really important data as we think about next steps in clinical development in Huntington's. We have engaged regulators about the next steps for development after these important data that we shared. We have alignment with the FDA following that meeting on advancing a potentially registrational phase 2-3 study with caudate atrophy as the primary endpoint. The really important work that was done by the community on restaging Huntington's disease from a research purpose, the new ISS staging of stage 0, 1, 2, 3, 4, the caudate actually is consistently declining through those stages. And so what's important about that is we think about the opportunity, as we are about our upcoming potentially registrational phase 2-3 study, where we're gonna look at caudate atrophy as a primary endpoint, means you can actually move much earlier in the disease setting. You can do that because the new staging criteria actually has stage 1 and 2 patients. And in stage 1, you have caudate atrophy occurring in the absence of symptoms. So we can use this as a biomarker endpoint to be able to look much earlier in the stages of Huntington's disease and being able to treat, but also having a measurable endpoint that we can follow to see about slowing disease progression. That's the opportunity really in moving the treatment paradigm for Huntington's disease earlier. The disease process starts early, but the symptoms take time to develop. And like we know from all neurodegenerative diseases, the earlier you can treat, the earlier you can prevent the loss of neurons, the greater the impact and delay to change in function later. That early treatment could allow people to potentially live a normal lifespan with full functional capacity. And I think that's the aspiration that all developers within Huntington's have of early treatments and what that potential could be on the other side.
Early treatments meaning we really need to make people in the community realize that there is no time too early for them to get engaged in this?
It's really important, but the impact that getting involved in studies earlier, what that could have in any capacity, whether it's in a therapeutic study, whether it's in a natural history study, just participating in research in general accelerates the pace that we can develop new medicines.
Engagement.
Engagement
of the
engagement of the community is critical.
Of younger people, priceless.
It's critical.
Researchers were also updated about other shots on goal that aim to lower Huntington.
ALN-HTCO2 is a RNA interference medicine that we've developed. Alnylam as a company has been around for more than 20 years, working to take the initial discovery in the lab that RNA interference is something that exists in all of us. It's a natural biological process that regulates gene expression. What we as an organization did in the early days was learn how to make molecules to co-opt or borrow that natural mechanism to point it at the specific mutated genes that we were interested in to turn them down or silence them. It's like turning off the spigot. And so once we figured out how to do that, Huntington's was one of the first places we wanted to go because the unmet need is so high, the target is so well understood in terms of the root cause of the disease. It's a very natural place to put this. The good news is that this molecule is not the first molecule we've put into clinic with this technology. We have a more advanced program targeting amyloid precursor protein, or APP, in Alzheimer's patients. And what we've learned with the platform was to get very, very long duration of silencing. After one intrathecal dose, we can see lowering of the target protein in CSS up to a year after that. Because it's so durable, after a single dose, we have very long-acting protein silencing or protein lowering. That will allow us to give very infrequent dosing in clinic. Over the lifetime of a patient, the total amount of drug that a patient has to take or put into their body will be much lower than a lot of other technologies. We initiated a phase 1 single ascending dose clinical trial late last year. We're working our way now through this year, adding additional patients, ramping up the dose, really trying to characterize the safety, the target engagement, understanding the durability of the molecule. And that information is really important to give us more confidence that it's doing what we expect, but also to allow us to pick the dosing regimens that we'll use in a phase 2/3 study that will come next to really test the efficacy of Huntington lowering with our strategy. We'll probably have our first real substantial data package to share next year at one of the Huntington meetings. And then if the data is encouraging, we're hoping to move directly from a phase 1 into a phase 2/3 study that would really test efficacy, potentially serve the basis for registration.
It's a really exciting time for Roche because we have multiple programs tackling Huntington's disease. We believe that by having multiple approaches in parallel, it's the fastest way to get treatments to patients. So we've been able to share clinical trial data from our previous trial generation HD1 and also from our observational natural history study. We're being able to share clinical data and MRI measurements. And this data can be used to understand how clinical measures change in a clinical trial. And that can be used for the community to enable drug development for everyone.
That's fantastic. Liz, you're another Roche product now, which is a different shot on goal, is it?
That's exactly right. So what we are developing is a one and done genetic medicine, a gene therapy, that allows us potentially to very potently lower Huntington in the most affected brain regions with a one-time therapy. This is a harmless virus, which we're able to use as a carrier for a microRNA that works to lower Huntington. It's delivered with a neurosurgery and then release the virus over a period of time into the patient's putamen and caudate.
So how is the tominersen trial going?
We're now fully recruited, so we reached our 300 mark in January of this year.
Yeah.
It's really been a testament to the community and patients and families helping us to get to that stage. So it's a really important milestone, and we now hope to complete the study by the end of 2026. So we're really excited.
Last year, we heard about the growing excitement around another approach to therapies called somatic expansion. There too, in the space of just 12 months, the field has grown exponentially.
So there are these studies called GWAS studies, which stands for Genome-Wide Association Study. What does that mean? It means they look at all of your DNA, so your whole genome, and what they say is, where is the variability? You know, is it green eyes, blue eyes, brown eyes, those kind of variable portions of different types of genes, and which ones affect Huntington's. So modifier genes are really important, and there's modifiers for all sorts of things, and we've just focused on those genes that are modifiers for the disease processes relevant in Huntington's disease. And from that, what they found out is there is one disease pathway that can either slow down your Huntington's or speed it up, and that pathway is part of a system called the DNA damage repair pathway. We think our DNA is kind of set in stone when we inherit it, but actually it's a bit of a war zone, your DNA. All sorts of things can happen to it. Too much sunlight, X-rays, chemicals, all sorts of things can damage your DNA. And you therefore need a whole load of processes in each and every cell that keep your DNA in good working order. And these are called DNA damage repair systems. And one of these is designed for correcting essentially typos that can occur in your DNA, where certain base pairs, so the letters that make up the DNA strand, can get switched for a different one, that it can be, cause disease. And so these processes are designed to stop that. And one particular branch, when it goes wrong, is what leads to this CAG repeat expansion. And that process, or that pathway, is called mismatch repair. And because we know this is mismatch repair dependent, it's that pathway that's at fault, we need to stop that pathway working. So as a biologist, we looked at all the different proteins and genes that make up that pathway and say, well, what's the best point of intervention? How do I make a drug that targets the right gene or the right protein that will be effective, so it will slow or stop disease progression, and most importantly, safe, so that it can become a treatment that everybody with HD should take. And we found a particular target, and the human genetics pointed to this, called MSH3, a gene that we think is really important in this mismatch repair pathway, and we think could make a fantastic treatment for HD. What we worked out is that we can target this with a small molecule drug. So what do I mean by a small molecule drug? I mean something that is a tablet or a capsule, a pill you can swallow with your breakfast orange juice and be a treatment. When you talk to HD patients, a pill they can pop in the morning is the ideal type of drug they would like. No invasive injections or surgery, and much easier. You don't need a fancy medical centre to take these kind of medicines. You can just go to your pharmacy and get your bottle of pills. So we worked that out, that type of drug could be made for this MSH3 gene. And that is different from the gene therapies or the ASO drugs that are being tested in the clinic at the moment. So we're at the stage where we have what's called a development candidate. So we are picking out that single molecule that we think is going to be safe and effective, and then taking that into those important safety trials in animals before we can then start clinical trials in people, hopefully in the second half of 2026, so just next year.
Wow.
Can we treat before people have symptoms and either delay when they will get them, or hopefully even prevent it altogether? And I think the answer to that is yes. And those GWAS studies that pointed us to this biology again support that idea, because what they did is they looked at people without any treatment and said, you know what, some of these people are going to get Huntington's 20 years later or 20 years earlier than the average Huntington gene carrier. And so we already know that there's a 20-year window we could potentially slow down disease by. So hopefully, with a really good somatic expansion inhibitor, we can slow it down by 20 years, maybe even more if you start treating when you're, say, a teenager or even as a child. And there's a really good medical analogy for this, of how that kind of evolution of how a therapy is used. If we look at the cholesterol-lowering drugs, they were first used in people who'd had a heart attack to try and stop them having another one. Then people tested people at very high risk of having a heart attack and showed it reduced the chance of them getting a heart attack. And then it went a step further back. Those who've got high cholesterol take this, much less likely to go on and get a heart attack. So Huntington's will do, I think, that same kind of stepping back so that we get to where we all really want to be, which is as soon as we know somebody is a Huntington gene carrier, we can put them on a medicine that's safe and convenient and hopefully stop them ever getting Huntington's.
Harvard professor Steve McCarroll's recent study has turned heads beyond the scientific realm as it helps explain the steps that determine the course of HD progression.
So, Steve, explain to me what it is that we have been discovering in the scientific world about the timing of the onset of Huntington's disease and what we've learnt in the last couple of years in particular.
It's always been one of the mysteries about Huntington's disease, why there's this long latent period where people are perfectly healthy for decades, often before they even begin to develop symptoms. And we increasingly think that what explains that is the DNA repeat that someone inherits genetically that ultimately leads to Huntington's disease is a certain length at the time it's inherited, most frequently in the 40s. But research over the years has shown that the length of that repeat is unstable. What's happening quietly inside the brain over the course of a person's life is that in at least some cells, that repeat is getting longer and longer. That's the phenomenon of somatic instability. And that was what we were trying to understand better. What the scientists in my lab did is we developed a technology to study each of the individual cells in a piece of tissue. And for each individual cell to recognize the length that the CAG repeat had reached in that specific cell, and then how that cell is using all of its other genes, what genes it's turning on, because from that information, we can tell what kind of cell it is, and we can also tell something about the health of that cell. And that was ultimately what allowed us to recognize the point at which the DNA repeat starts to undermine the health of those cells. So these experiments taught us that this plays out in the following sequence of events. So in the first phase, which takes many decades, a neuron's perfectly fine, but its DNA repeat is slowly expanding. Let's say that someone inherited a version of the gene with 42 CAGs. It may take many decades for that to expand to 80 or 90 repeats. When it reaches 80 or 90 repeats, this expansion starts to pick up speed. And so it's taken many decades to reach that point, but then it may expand from 80 or 90 repeats to 150 repeats in a few years. But it's only at that point that the gene even begins to become harmful. And so a neuron, we think, really spends 95% of its life with an innocuous Huntington's gene, a gene that's innocuous but unstable and slowly expanding and kind of a ticking DNA clock. The kinds of neurons, of nerve cells, that are most vulnerable in Huntington's are exactly the cells in which the repeat is expanding the most quickly.
Right, and what are they called?
They're called striatal projection neurons, or also called medium spiny neurons. They have a variety of important functions. They control movement, but they also contribute to cognition and motivation. Now, this ticking DNA clock, this process isn't happening in the same rate in each individual neuron. So we can see in the same person's brain both neurons in which the repeat has expanded a lot, and those are neurons that have begun to experience biological changes from that. And then many other neurons in which the repeat has not yet become toxic. There's this beautiful human genetics work that's been done to identify genetic modifiers of age of onset. And very many of those modifiers are in genes that seem to regulate the rate of this somatic expansion of the CAG repeat. And one of the insights from that work is that our own DNA repair enzymes kind of contribute to this lengthening process. One implication of that, though, is that it might be possible to slow the activity of those enzymes in a way that would slow the rate of somatic expansion. And so in a given person, it takes on average, let's say 50 years for the repeat to expand from 40 to 150 repeats. If you could slow that process by 50%, it might profoundly postpone the onset of any disease symptoms. That's something that we're excited that many drug companies seem to be getting enthusiastic about working on and developing substantial programs to try to do.
Because if that can happen, that actually, in a sense, is something that wouldn't need to necessarily happen at birth or as a young person. This could be something that could just be introduced, importantly, before the major expansion happens?
The sooner you treat, the better. But one of the surprising things that we learned from these experiments is that even in someone who already has Huntington's disease symptoms, that most of their neurons still don't yet have a toxic Huntington gene. Wow. So it might be that even in someone with Huntington's disease symptoms, that it's possible to stop or delay progression of symptoms if one could stop somatic expansion.
Wow, that is exciting.
There's been really smart therapeutics work on Huntington lowering, and then somatic expansion opens up a new front on the problem. And I think one of the things that these experiments taught us is that there's much more therapeutic leverage in somatic expansion than we thought. That may be a really promising place to target, because for most of a neuron's life, it doesn't have a toxic Huntington, it just has a ticking DNA clock. And so we're really excited that many pharmaceutical companies have expressed interest in this. We actually started sharing these results at this conference 2 years ago. It's been exciting over that time to have a lot of interactions with people from biotech and pharmaceutical companies who are starting programs in this area.
And you know, I know that the expansion of the number of companies here has been so exponential anyway, but I mean a very large percentage of that are people involved in this field.
Yeah, I think they sense that there's a real opening here.
Yeah. Everyone who comes from a Huntington's family has been aware of the fact that you don't seem to get sick at all for a long time, but then they go off the cliff.
Yeah.
Is this the explanation for that?
We do think it is, yes. So you use the cliff analogy there, and I think this is really a good one for thinking about what does somatic expansion mean? Because the Huntington protein bit where it's toxic is where you've kind of fallen off the edge of the cliff, and it's that downward trajectory till ultimately each individual brain cell will become more and more dysfunctional until it dies. But what happens before you get to the cliff edge? You're not in free fall at that point. And the somatic expansion process is what moves your vulnerable brain cells to the edge of the cliff and eventually pushes them over. So if we can slow down somatic expansion, you don't get to the edge of the cliff and you shouldn't see any symptoms. And we may find that somatic expansion is probably best at stopping your brain cells running up, towards the cliff, but Huntington lowering is the parachute that slows them down if they've already gone off the edge.
Okay. Does this in any way give the prospect, even a remote possibility that people who are already symptomatic with the disease might be able to have some way of affecting it?
So I think a really, really critical study, the work of Steve McCarroll and that team, and what they showed is that it is really a cliff edge approach. Your cells are fine, even if they've got a Huntington gene that's in the disease range, and even if they've had some level of expansion, it's not till they reach the cliff edge that they really start to go wrong. With a cliff edge, as long as the brain cell hasn't gone over the edge yet, I think it could be entirely rescuable. And very few cells we've found are actually in that free fall off the edge of the cliff at any one time. So if we treat a patient who's already got symptoms, sadly I don't think we can get back brain cells that have died, but I think what we can do is stop any more going over the edge, and so stabilize the disease and prevent it getting worse.
Pioneer of ASO therapeutics, Don Cleveland, gave the distinguished scientist address. He has seen the field evolve across more than 4 decades.
Do you think that it's remotely possible that we would be able to at some point recover neurons?
Neuron recovery is one of the most ideal situations for which we just never really had evidence in the aging nervous system that you can generate new neurons. But we do have that evidence today. We can make new neurons. There are sites in our brain, 2 of them, where neurons are normally born, but they stop being born in adult life. But the precursor cells are still there. And if we could just nudge them into rejoining the pathway to become neurons, that could really work. And we have demonstrated that in mice, we can do that for striatal neurons. We certainly can test how broadly it's true that we can generate new neurons by waking up dormant precursor cells that all of us have in our brains, but they're just dormant.
So Don, how close to an effective treatment are we?
We certainly are well on the way to having scientifically and medically sensible ideas moving forward to clinical trial. There are some in the final phase of trial. I'm optimistic. We need the proof, of course, the outcome of the trial, but that's why multiple trials are exactly what we in the community and the patients and their families, exactly what they should want. And that is what's happening. At the CHDI meeting in Palm Springs this year, I heard additional ideas. I was not familiar with them, but the evidence for those therapeutic approaches is very credible, and they should be pursued in parallel, hoping that actually multiple ones may have a benefit, and then combinations of them could be truly transformative in the therapeutic approaches. And I think that we have multiple shots on goal is exactly what you want. I think that in half a decade, we will certainly have the outcomes of those that are in trial today. And I think that there's good reason for optimism that there will be one or more of them that prove to be quite beneficial to patients. Success, at least a partial success, will be likely in the next 5 years.
The sheer number of approaches being pursued in clinical trials has given researchers here the genuine belief that we could be at a tipping point. Now, more than ever, all of us families need to engage to drive forward this groundbreaking research.
Waiting for a golden age with a garden in the sun and a shadow on your face. No, living like we're on the road, all hiding from the end, but it's already begun. Oh no.
So I'm really excited to see really the flurry of multiple shots on goal now that's really moving forward, and it's very exciting times right now. Must be very exciting times for the HD community.
It is.
Yeah.